Talking about pathophysiology of the cell and cellular injury. Cellular injury occurs when the stress on a cell exceeds the cell's ability to adapt. Remember, whenever stress is placed on a cell, the cell will respond by growth adaptations if it's within its ability to adapt. If it's within its ability, growth adaptations such as hypertrophy, hyperplasia, and atrophy, and metaplasia and dysplasia, those will occur. However, if the stress exceeds the cell's ability to adapt, then we will have cellular injury. You have two types of cellular injury: reversible, which means that you can reverse the effects of it, or irreversible injury. And with irreversible injury, that's what can lead to cell death, aka necrosis. Hypoxia is low oxygen delivery to the tissues. Once again, hypoxia is low oxygen delivery to the tissues, okay? That's very different to ischemia, and it's very different to hypoxemia. Hypoxemia is low oxygen dissolved, low partial pressure of oxygen. That means low oxygen dissolved in the plasma. Partial pressure dissolved. O2. Hypoxia can be as a result of different factors, but mainly these three. Also, in terms of cellular injury can be caused by these five: hypoxia, ischemia, and nutritional deficiencies such as vitamin deficiencies, trauma, or genetic mutations. And then hypoxia, as I was saying, is hypoxemia, ischemia, as well as a decreased oxygen carrying capacity. These two I've explained, or this one is self-explanatory, these two are fine. What about ischemia? Ischemia is actually decreased arterial blood flow through an organ. So ischemia is decreased blood flow through an organ, whereas hypoxia is low oxygen delivery to the tissues. Ischemia can be due to a lack of arterial perfusion or lack of venous perfusion, decrease in arterial or decrease in venous perfusion. Okay, why is hypoxia so important? Well, in hypoxia you have oxygen, and oxygen's your main guy. Oxygen has a very important function as the final electron acceptor in the electron transport chain. Without this final electron acceptor, there will be decrease in oxidative phosphorylation, which is our money withdrawal, which is our ATM for our cash. Remember, ATP is our currency in the cell. So if oxidative phosphorylation, our cash withdrawal, is not working, we're not going to get ATP, and we're not going to be able to carry out our major cell functions. And that is what's going to cause all of these reversible injuries. Remember, if there's a block in the arterial end, that's going to be a decrease in arterial perfusion, and if there's a block on the venous end, that will be a decrease in venous perfusion. This box here summarizes quite well reversible injury and what causes it. Basically, if there's no oxygen, the mitochondria won't be able to have, when it's in a mitochondrial matrix, the electron transport chain won't be able to function properly, and there will be less oxidative phosphorylation, less ATP, which will then can eventually cause cell damage. Less ATP means three main things. Less ATP means, firstly, start from the bottom, work our way up. Firstly, our sodium-potassium pump, which is ATP dependent, will not work as well, and as a result, sodium will accumulate inside the cell. Accumulation of sodium, wherever sodium goes, water will follow, will cause water accumulation too. Water accumulation is also known as cellular swelling. Now, if sodium is moving in, because this pump over here is not working, so will water move in. If water moves in, this will start swelling. If this starts swelling, the microvilli that were first finger-like projections that were very motile and functional, they will start to get ruined. Loss of microvilli is your first hallmark. Well, your hallmark for reversible injury is in fact cellular swelling. After that, as a consequence, you have loss of microvilli and basically these outward bulges of water called membrane blebbing. What about the rest? Well, as a result of lack of ATP, you will not be able to do aerobic glycolysis. You will have to go to anaerobic. And as a result of going to anaerobic glycolysis, you're going to have a build-up of lactic acid, which is going to decrease the pH, which is going to do something very bad, such as denature our proteins, and that's going to wreak havoc in our cell. Thirdly, the cell wants to keep a low level of cytosolic calcium. But what is going to happen in this? Your calcium's going to increase because you have a calcium pump which is ADP dependent, and that's going to stop working. Calcium's going to stay inside the cytosol, it's going to build up. And what does calcium do? Activates enzymes like proteases and endonucleases. Endonucleases have the capacity to break down DNA. So that can be really bad. But also something I forgot to mention, loss of microvilli, membrane blubbing. But if your cell is expanding, what do we have inside the cell? RER. The rough endoplasmic reticulum will start to expand and swell, and the ribosomes are only loosely attached to your RER, so the ribosomes will disperse and disassociate. In terms of your irreversible damage, I just want you to remember the word PALM. P for plasma membrane damage, L for lysosomal membrane damage, M for mitochondrial damage. Plasma membrane damage will cause cytosolic enzymes to leak. This will cause a high serum level. That's how when we test for myocardial infarction, we can check for serum levels. Lysosomal membrane damage will cause hydrolytic enzymes to leak out. And then calcium can actually react with those enzymes too. Finally, mitochondrial membrane damage will cause complete loss of electron transport chain and cytochrome C to leak as well.